Fantastic. I can see that we have people joining us. Hello, welcome everyone. Great. Well, let's make a start. So I just want to say hello and welcome to today's open event, where we will be looking at our programs in genetics, multiomics, in medicine and drug design. Thanks very much for joining us. My name is Kate Thomas. I'm the Faculty Events Officer for the Medical Sciences faculty at UCL. We're really hoping this session will provide you with an opportunity for you to address those course-specific questions you may have, and will help you to gain an insight into what it is really like to study in our division of medicine. So this afternoon, Horia and Edith will provide us with a summary of our exciting postgraduate programs. And there'll be a Q&A session and plenty of interactive opportunities for you all to join in throughout today's session. This session is being recorded and will be made available on our website following today's event. We're here to respond to your questions, so please do share those throughout using the Q&A function on Zoom. Now to introduce our speakers. We have Horia Stănescu. Dr. Stănescu is a specialist in medical genetics with more than 20 years international experience in biomedical research. Having spent 4 years at the National Human Genome Research Institute in the USA, he joined the University College London in 2007, where he is now currently a lecturer in renal genetics and a primary PhD supervisor at the Faculty of Medical Sciences. And Edith Chan. Dr. Chan has over 17 years of pharmaceutical and biotech experiences in the area of molecular modeling, cheminformatics, structure, ligand-based drug design, and biocomputing. She joined UCL in 2009, and her research is focused on computational drug design. Thank you so much both. Before I hand over to our speakers, we would like to run a couple of polls. Please do answer those, and I will just launch them shortly. So the first poll is asking, which best describes you? The options are healthcare professional, biological sciences graduate, mathematics graduate, computing science background professional or other. And the second poll is, what course are you interested in applying for? Is it MSc Genetics and Multiomics in Medicine, or MSc MRes Drug Design, or even if you're not sure yet? Let's have a look. I'll just share those results quickly. So we have about a good mixture actually. So 62% of you are biological sciences graduates, 23% healthcare professionals, and 15% other. And again, a mixture for the courses you're interested in. 31% are MSc Genetics and Multiomics in Medicine, 46 MSc MRes Drug Design, and 23% of you are not sure yet, which is completely understandable. We know that it is a big decision, so we're hopefully this event will provide you with all the questions you may have to help reassure you which course you want to apply for. Fantastic. Without further ado, I'd like to hand over to Horia.
Thank you very much, Kate. I'll share my screen. I will give a short presentation, starting by defining the terms in the title of our program. And hopefully you will get a feeling about what, how, and why we are doing, and what we are doing things the way we do in our group. So in the context of medicine, what we are trying to do is to understand the way diseases originate. Because if we want to diagnose and hopefully have a chance to treat disorders, we want to understand the mechanisms of development of a disease. And this is the realm of pathogenesis, which comes from the Greek words genesis and pathein, pathos. Genesis is a Greek word that is also related to inheritance, and genetics is the science of heredity. And genetics combines interesting and paradoxical terms, constancy and variability. And if we are to illustrate variability, a beautiful depiction of variability would be a coral reef, for instance. And a coral reef shows how beautiful complexity can be. And if we are to hope to understand complexity, a way to tackle that is by decompose it. So one of the components of a coral reef is the red coral that you see here. And the red coral is also suggestive for what Charles Darwin called the tree of life. Well, he didn't call it the tree of life, but the shape is very similar. And nowadays, we would rather refer to the tree of life as being the network of life in order to suggest the way complexity is created and information, genetic information, flows along and across the branches of the tree of the network. And if we're talking about genetic information, well, genetic information, the information of life, is encapsulated. Their material substrate are molecules which are called cementioids. And cementioids are the different types of macromolecules that carry the genetic information. And those molecules are referred to in the central dogma of molecular biology. The central dogma of molecular biology was stated in the 50s by Francis Crick, which you probably have heard of from the famous Watson and Crick duo, which is part of the larger team that solved the structure of DNA, which is one of the cementioids, in 1953. And just to mention that the central dogma, which tells us that DNA is transcribed in RNA, being later translated into proteins, these three being the classes of cementioids. So the central dogma was first stated at UCL. Francis Crick being one of the alumni of UCL. And what the flow of information, of genetic information, does is to bring to light, to express the beauty of biological phenotype. And what a phenotype means is just the way the individual appears. It's an old concept stated by Johansen in 1909. And from phenotype, there are a couple of other derived concepts, which is the phenome, the complete phenotypic representation of the species, not only of the individual. And this is why phenomics is the science that investigates the phenome. So to bring to light, as the painter brings to light, in this case, a body, this is Rembrandt's anatomy lesson of Dr. Tulp. And this is an example of studying the phenotype. And what we see here is Dr. Nicholas Tulp in the 17th century dissecting. So what we see here are hands dissecting hands. And this is interesting. In the next image, we can see hands drawing hands. And this is just to suggest that the human body is a very interesting instance in which we have the object and the instrument of research. So our bodies are the target of our research, but our brains are what makes research possible, our brains and our hands. And staying with the theme of hands, this is yet another picture of hands to show you that when we are referring to phenome, that is to bring to light, we can sometimes refer also to invisible light. In this case, electromagnetic rays in the X-ray spectrum. And this is to suggest that phenotyping is very complex and relies on technology and subtle and multiple methods. Now, X-rays are also used, have been used, to solve the structure of the semantide that I earlier talked about, that is DNA. And this is the famous so-called picture 51, the first picture of the picture that was used into solving the structure of DNA. And this was a picture obtained by Rosalind Franklin. And Rosalind Franklin, together with Maurice Wilkins, were, as I said, part of the big team that solved what they called back then the secret of life, the structure of DNA. Now, from the structure of DNA to the secret of life, there is a long way. And partially, this way relates to the information that is encapsulated in the DNA molecule, and that is sequence. And we were first able to read the sequence, the entire sequence, encapsulated in the long DNA molecule in 2003, 50 years after the structure of DNA was solved. And this was possible due to the Human Genome Project. And the Human Genome Project represents a shift from the approach of classical genetics, in which we were studying one gene at a time, towards genomics, which means now we have access to the complete genomic genetic or genomic information, that is 20,000 genes in the case of the human genome, and we address them in parallel. Just to make the distinction even clearer, the way we study classical genetics is to study families, and here we have an example of a pedigree, a family tree, in which there is a trait segregating, and we study by linkage analysis the way the trait is segregating in this tree. In genomics, we are tackling usually more complex phenotypes, complex disorder, and this shows you a so-called Manhattan plot, in which you can see on the x-axis the entire genome with the numbers corresponding to chromosomes, and on the y-axis is the significance of the signal which represents the association of the trait with a given position on the genome. So that's the type of analysis that we are performing in order to understand what is the genetic origin of a disorder. But if we are to understand disorders, and our lab is mainly interested in kidney disorders, this is why I'm showing you the phenotype of a kidney disorder. Here you can see polycystic kidneys at different methods, CT and ultrasound, and at different resolutions, macroscopic and microscopic. But in order to understand what the link is between an alteration in the genetic material and the alteration at the level of the phenotype, we need to understand how DNA is expressed in RNA, and we need to see the difference between RNA expression in normal individuals and in affected patients. And that is the realm of transcriptomics. Transcriptomics is everything that is related to RNA. It means the study of the transcriptome. And the transcriptome is the complete set of ribonucleic acid molecules. So again, the idea is that we are studying complex sets of macromolecules of cemantides in parallel. And this also represents an increase in complexity because you're moving from 20,000 genes to about 100,000 possible transcripts of or messenger RNAs. And the next step in complexity that we are tackling when trying to understand diseases, and this is an example of a different class of diseases because I told you we're interested in kidney disorders, but we're also interested in neurological disorder, and this is an example of a neurological disorder. And the next level of complexity is given by proteins. So here is a three-dimensional depiction of a protein, and the alteration at the level of the structure of the protein leads to the problems in the patient. And in order to understand this alterations at the level of proteins, the science is the science of proteomics, which is very ambitious and has as a goal the characterization of all proteins in a living system. And as I keep insisting, our approach is of the parallel investigation and interpretation in a very large number of proteins, ideally all of them at once. So this was a very fast-paced walk through the chapters of multiomics that I hope I was able to convey the shift that is happening nowadays, the shift from classical biology, which has an emphasis on small-scale modeling and single molecule experiments, as I said, classical genetics, one gene at a time, towards a systems biology approach, in which we are dealing with large, complex sets of data, and we need to deal with them by using powerful tools. And one of the most powerful tools is, of course, the computer. This is why our program is very heavily computer-centered, and it has two modules that are dealing with bioinformatics and computational biology. So these are the modules of our program. There are 8 modules, and added to the 8 modules, which are genetics, then the core multi-omics modules, which are genomics, transcriptomics, proteomics, and phenomics, which I hopefully was able to shortly define. The 2 computing modules that are very important in terms of being able to analyze complex data sets with powerful tools, and very important, the ethical, legal, and societal aspects of multi-omics. And added to the 8 modules is also a research project that you'll be asked to perform. Each module consists of about 10 chapters. Usually it's a lecture in the morning and a practical session or a seminar in the afternoon. They are taking place on the Royal Free Hospital campus of UCL. And what we try to do in this program is to give you a wide perspective of biology. We are more focused on concepts than on techniques. Of course, there is the technical and practical aspect, but what we want you to take home is the concepts, the deep understanding of what we are studying. And in order to do so, we have an integrative approach. We are relying on the history of science. We have philosophical discussions, and every now and again, of course, we try to spice it up also with references and metaphors from arts, as I was showing you some paintings, for instance. Our approach is very collaborative. We are hoping not only to teach you, but also to learn from you. to get interesting questions and vivid discussions. And also we want to teach you the fact that cooperation is important in biomedical research. It's very fruitful to get computing specialists collaborating with biologists, clinicians collaborating with researchers, and theoretically inclined individuals collaborating with people who are handy and like to perform experiments, of course. And I'll end my presentation with just a handful of pictures with some of my colleagues. A couple of them are in the room right now and are going to take some of your questions later in the question session. Thank you very much.
Thank you, Horia. Now over to you, Edith.
Okay, hopefully you can see the slides. If not, just let me know now. So
I can't see your slides, Edith. So.
Okay. Is it okay now?
Not yet.
Still not seeing them yet.
Okay.
Yep, I can see them now.
Okay, so this is the MFC MRes drug design program, and I'm the program director, Dr. Yves Chen. So I have some images here. So our program is based in the Cruciform building. Is one of the historical building. It has 200 years old history, and it was a Victorian hospital before. So if you join us, you are joining part of the history. So before we even begin, let's watch a movie. So what is the idea of drug design? What you're looking at actually is a protein structures. And usually it has a binding site where it will have interaction with a drug molecule. Should we see it again? So, so our ultimate goal is we can design a drug that can occupy or inhibit that binding site and be a successful drug. Okay, so the course is not just about like design molecule, because before you even starting to do, oh my God, what do I do? There's so many things to learn. So for example, the drug design is our ultimate target. And it doesn't necessarily that only include the conventional drug ideas that you have, like small molecule. It could be new therapies. So we'll be talking about vaccines, antibody, small molecule, siRNA. And then on the other side of it, you have to have all these other science that have to support this. So you'll be learning structural biology, X-ray, NMR, a lot of computational design concept, molecular modeling, and a screening technique, a lot of things that basically involve in the drug discovery process, and you have to do for the drug design. And at the end, we will also learn something about the commercial aspect of drugs and also the intellectual property, because at the end of the day, we are not trying to design aspirin or anything like that. It has to be novel and effective. So like I say, besides the traditional things, we talk about antibody, CRISPR, Cas9. So these are the new therapies in drug discovery. Small molecular drug design and molecular modeling and cheminformatics. Now, our curriculum changes a little bit every year to respond to the new technology or concept in drug discovery. So artificial intelligence, machine learning have been introduced a couple of years ago. So we'll adjust the topics a little bit every year. So the more classic ones, structural biology, NMR, X-ray, we have to learn it. Also, in our big discovery world, we have to learn how to handle biological data, especially big data set too. Intellectual property, the basis of pharmaceutical company. And so we also have a lot of cutting edge lectures that we invite people from industry, big pharma or biotech to come to tell us real case studies, not just concepts also. The organizations of the program are as follows. There are 8 top modules. So basically 4 modules per term. So from September to December is 4 modules. And then January to March is another 4. And then after Easter, usually you'll be engaged in the research projects. The research projects are really diverse in our program. It could be a wet lab, it could be related to chemistry or biology. It could be dry lab, computational modeling, programming, and usually you have to do at least 12 weeks and 60 credits. And then in August, then you will just finalize your dissertation. Now, also that we introduce the examinations almost immediately after each module is taught. The reason for doing that is then when you have the knowledge fresh in your head and you just do the exam. This is actually a very good organization because all students almost, because then you finish all your exams before March and then you can concentrate totally in your research project. Usually that is the best part of the program. Like I say, our program is based in the Wilson Institute for Biomedical Research, which is at the Crucible building. All our teaching are on site, and we have an advantage. We have the Crucible library and the hub. We have a lot of computer classrooms there. Also, a space for student discussion is just in the basement of our building. Wi-Fi, of course. And then upstairs, we do research there, so 12 laboratories. And besides just learning the concepts and the practical of drug design, there are also other skill and training courses that offer to you, like, for example, presentation skill, writing skills that almost everybody should have before they leave university. What do our students do afterwards? So again, just plenty of options. So the majority of students, they go on and do a PhD in different university. UK, I have students that went to Oxford, UCLIC. The world, people go to Germany, Italy, USA, or Taiwan. Jobs are also very diverse. Some people do research assistant in laboratory, either in university or pharmaceutical company, teachers, sales, marketing, and yeah, it's just very, very different. So so if you ask me who has become a big boss at the end, actually I have one student who is a VP in AstraZeneca. So how good is that in marketing? So you could have publication chances too. I have seen people doing a 3-month project that they can publish. And so the opportunities are all here, but you just have to do grab it and go for it, okay? So I give you details of my web link there. So if you have more questions, you can look at that. And if you have questions today, you can ask me too. Okay, thank you.
Thank you, Edith. So yeah, if everyone has any questions or anything about both our presentations today, please do use the Q&A feature. I know that a lot of these events we've had questions around the learning and what will teaching potentially look like in the current climate. So I don't know if Edith, if you would like to explain a bit more about the teaching side of things, that would be great.
Of course, the last 2 years, because of the COVID, all the teaching, so the first time that we locked down, so we have basically totally online teaching. And then the second time we have a little bit of online and sometimes face-to-face, but at the end it's mostly online. But looking forward, and the experience was really good for students. I mean, all of them were very motivated, and we all everybody got their degree, no problem. But moving forward, 2022 onwards, everything will be face-to-face. I think you guys all want that. So yes, so 100% face-to-face, that's what I would say. So you will have a complete on-site student experience.
Fantastic. Horia, did you have anything you wanted to add, or is it similar situation?
It's our program. It's only the second year. It's a new program. So indeed, we had online teaching, and now we are having also face-to-face teaching. And we much prefer, of course, face-to-face teaching. We prefer having students in the lab and interacting with them, and we're looking forward to normality.
I think that's all received with welcome open arms. So perfect. We do have a couple of questions that just come in. So first one is from Naomi. Thank you, Naomi. Can you give some examples of previous research projects? So I think that has been a question for both of you, as there's not a course specific tied to that one. Edith?
Yeah. So like I say, there are many. So I give you some example in different kind. Like chemistry project is medicinal chemistry. It could be involving people synthesizing compounds and then testing them in biophysical assay or biochemistry assay. So this is called a medicinal chemistry project. There could be purely biological related projects, screening projects. So basically you might be using a technique to screen a pool of compounds for potential drug possibility. So you could be doing crystallizing a drug target protein, yeah, X-ray crystallography. And it could be maybe setting up a biological assay for further usage. So just so many, because the reason is you are at UCL, you are in the Division of Medicines. We have over 100 principal investigator, and think about how many different projects you can get. And also every year we compile a big list for all the students in the divisions, so you can have a look and approach different people and get the project that you want. So getting back, so for theoretical or computational project, it could be doing Python programming to do machine learning. It could be look at a big data set, million compounds or data point, and sort out some maybe genetic profile, things like that. It could be doing more conventional dynamics project, look at how drug molecule behave in a protein target. So again, because if you come to UCL, you're lucky. Our research is just have so much opportunity, really.
That sounds very exciting. Thanks so much, Edith. And Horia, did you have any examples?
As I said, this being a new program, we don't have yet examples, but we have, as Edith said, we have a large spectrum of possibilities, everything from the wet lab. This is why we are covering genomics, transcriptomics, proteomics. So the idea is for us to get to know the students a little bit, to see what they're interested in, and then if they're interested to be in the wet lab, they can for instance, look for differences between normal samples and samples of patients with different disorders by the means of differential transcriptomics or proteomics, 2D gels, mass spectrometry. These are experiments we run in-house, and we also have collaborators that would be willing to take you on for the research project. And in terms of computational projects, indeed, there's the possibility to have large data set analysis, family tree analysis to look for a variant that is potentially causal in a disorder. That can be, as I said, either family-based or association-based, genome-wide association studies, genetic risk scores, modeling. We can look for the impact of potential genetic variants at the level of the structure of the protein that is similar to what Edith just beautifully showed you in her presentation. So there is indeed an immense spectrum of possibilities, and the moment we get to know what you're interested in, we can start discussing and see what is most appropriate for each and any of you.
Fantastic. So we have got a few other questions that have come in, so thank you everyone. So we have a question from Amelia, and this one is for Dr. Chan. So Amelia is wondering what qualities and skills from undergraduates you are looking for in students.
Yeah, so I think in general, we are looking for a student who has achieved better than 2-1 to be admitted to our program. And if you have a background in biomedical science or just natural science or mathematics, we welcome you.
Fantastic. And have another question, which is for both of you actually. So is there any advice for undergraduate students looking to apply for your programs? So I know that Edith, you probably highlighted those, but Horia, did you have anything you wanted to add of what makes a good applicant?
Well, of course, I agree with Edith, and I believe that the main quality that we're looking for is curiosity. I mean, we want people to be driven and innerly motivated. They should be here if they really want to learn and follow their drive to communicate. So openness is appreciated. Communication, as I said, we are a very collaborative team. We believe in discussion, in vivid discussion, and Socratic teaching, as we call it. So it's question and answer, and it's generally a very interesting intellectual atmosphere that we're dealing with, and we're hoping that each of you will contribute to that.
Fantastic. And we have a question that's just come in from Judy, so thank you, regarding drug design. Are there any differences between the taught degree and research degree in terms of future career path and opportunities?
My answer is no. I mean, it really depends on the individual performance. And I have students from either degree to get on to do a PhD or look for any jobs they want. That's really not, I think there's a myth that people think that MRes can enable you to find job easier, and it's not really. It's really dependent on individual performance.
Okay, fantastic. And there's always a question that comes up quite frequently actually within these types of sessions, but I know our audience are usually very interested in your story. So particularly what inspired you to go into your current careers? I don't know, Horia, if you wanted to go first.
Well, I started in a clinic, and I was working with pediatric patients in a neurology clinic for kids. And in pediatric neurology, the vast majority of cases have a heavy genetic component. So in order to understand what was happening to the patients, to try and help them, I was more and more driven to genetic dissection and genetic analysis. So then we wanted to set up a laboratory. I traveled to the United States in order to learned some of the modern technique for genetic analysis, and the rest is history. I ended up in research, and I never regretted that.
Thanks. And Edith?
I graduated in Cornell University with a degree, a PhD in theoretical chemistry. So I know a lot about molecules, I mean, so it's totally not drug related, yes. But I guess I was lucky. My first job was when I applied for a job immediately, I got a job in a pharmaceutical company doing, they call me a drug modeler at that time, but I knew nothing about it. But once you're there, you learn it yourself. You talk to your colleague. You learn on the job basically, and then I was so happy that I can apply my chemistry background into a big discipline like this. And so, like I say, I work mainly in big pharma. I have pharmaceutical company experience for 17 years. So by that time, you probably know everything about how it works. And then after that, I had an opportunity to come to London to join a startup biotechnology company that actually was a spin out from UCL. So I came here, so it was a startup. We have to invent and do a lot of novel discovery for the industry. And then after that, I also had another opportunity. I can join UCL. And so I joined UCL about 13 years ago. And I must say that academic, I've never thought about that I could go into academics. But since I have been in UCL, I really appreciate the freedom of research. And I think I have more ideas and I can exercise on them as much as possible that I want. And I mean, it's not like so much inspiration. I just want to tell you some of my career path.
No, it's really interesting. And a drug modeler, that's a fascinating title for a job. So fantastic. So we have got a question in the chat about just saying thank you so much for an amazing lecture. And they want to ask, so I think they have a degree in chemistry, and whether that is sufficient for them potentially to apply for one of the courses. But please correct me, Anis, if that's not right.
Yeah, that's fine. Yeah.
Yes, same for our program. So we welcome diversity. So biological background, chemistry background, physics, mathematics, computer science. We need all the help we can get in order to do gene hunting.
Perfect. Just having a look at the chat to see if I've missed anything or the questions. But yes, everyone, please do feel free to continue. I saw that a lot of the attendees today are biological sciences graduates. That's really interesting to have you on board with us today. Let's take another look through. Equally, if anyone has any questions about the experience of living in London and why coming to London particularly is the best for these courses. I know obviously Horio is currently sat in the Royal Free, so you've got a lot of different locations across London that you'll probably find yourself at, which is quite exciting. Okay, I think I've just got a question in. How do overseas students adapt to the programme and go forth after graduating? So I think that's a question to both of you.
Okay, so should I answer it first? Yeah, UCL has a lot of international presence. And so it is very different than from different country. So a lot of them go back to their own country and they found job easily. No, so sorry, that's not the question, is it? Yeah, so I go back, sorry, it was in Korea. So yeah, so in our, so I'm talking about all the program in the division of medicines. To help students that have from international background, we have writing courses that can help you to, because academic writing is so important. So we have English courses that you can take to improve your English and also to help you learn how to do academic writing. The division also provide a training course for presentation skill. So even though you are not a native speaker, you can we can give you some help on how you can express yourself or do public speaking. So there are a lot of these little things is optional, that if you feel like that you are not so good in some area, you can pick up those training courses. And in terms of learning, I mean, all our program have different type of learning. So it's not like you have to be a super duper English speakers, but all of us should have a basic standard anyway. So it will be quizzes, like lectures and things like that. So I think it's not new for us in UCL. I mean, we have a lot of things position that can help international students.
Fantastic. Horia, do you have anything to add?
It is indeed a very multicultural environment and friendly, and the laboratory has people from America, Asia, Africa, Europe, and this is actually part of the fun and exchanging ideas and exchanging ways of tackling problems and all the discussions. So they usually adapt. It's a friendly environment. It's I don't see any problems. And as you said, the overall environment of London is extremely multicultural, extremely tolerant, extremely welcoming. It's a good place to be, London and UCL within London.
Fantastic, thank you. We do have a couple of questions that have come in. So this first one is actually for both courses. What level of knowledge of coding / computing is expected prior to starting the MScs? So I don't know, Edith, do you want to start off?
Another question, so I'm going to answer both at the same time. Zero,
of course.
You don't need any, that's the point. I mean, like we have, even though you are both 21, I think, when you come in, you are from different backgrounds. So, but if you don't have any programming background, it's fine. So for us, for drug design, we have actually, I have a old colleagues from Glaxo Wellcome, GSK. He's going to come in to give a lecture to talk about AI and drug design and machine learning. So that's one way. We also teach basic Python in cheminformatics and bioinformatics. So we make it so that in 2 hours you will learn how to do Python. How about that? So don't worry about it, really. That's
a good crash course, 2 hours. Aurea, did you have anything to add?
Same here. We're actually sharing the bioinformatics module with Edith. So thank you, by the way, Edith, for the collaboration we're having. And indeed, we take everyone from zero. We start with basic Unix, Linux commands. And even if you were afraid of command line programming, we'll take you by the hand, show you step by step, literally from zero, how to find your way in an operating system, in a computer. We're talking even a little bit about the morphology of the computer. What is a network? What is an algorithm? And increase gradually the knowledge that is needed. So no worries in that respect.
Yeah, can I just also say something? But it doesn't mean that the drug design course is about machine learning and programming. This is just part of the so many things that we teach. So if you hate programming, you can still come and learn other things too.
Hopefully that answers both of those questions. So thank you both for that. We have got around 9 minutes left. So if anyone does have any other questions about either courses or studying at UCL, we have got a bit of time left. So feel free to drop any of those questions in the chat or in the Q&A section. And Edith and Horia, how are you finding it now? Obviously, we've gone from everything being remote, and now it's great that we're welcoming students back on campus. Are you finding that there's much more of a sense of community being created, and actually seeing students in person's really, really been a positive things for the student experience, should I say?
Yeah, definitely. Because also usually all our programs have group activities. So now you can see a group of students like talking to each other. That is a very good sign, because collaborations, working in a group, team spirit is quite important.
Yeah, definitely. And I think from your picture, the lovely photo that you shared, that showed that quite nicely.
Yeah, we haven't had a party yet, but I think next year we will.
Let's hope, fingers crossed.
Yes, that's the way to create team spirit indeed.
Perfect. I think we have one question that's just come in. Do the programs collaborate with other departments in terms of research?
Absolutely. Like I say, that you can pick anything you want to do with other people. And the course, I mean, it's not just taught by me. I mean, I think I only do 4 lectures. All these other people are from external, from internal, from division of medicine, from another department, from like bioscience, genetic. I mean, yes. I mean, I think that is also why UCL is so good in research, because our collaboration is really, really good.
Exactly. Same here. I mean, most of the lecturers on the course are from different backgrounds, from different places, even from different countries. We're having a collaborator from Italy. We're having a collaborator from Germany, from all around London, from the Crick Institute. So, and they all have different ways of tackling problems, different philosophies. There's a lot of freedom, a lot of ways to choose one's way.
Fantastic. And we haven't, I think we've answered everyone's question. Just having a look now. So yeah, I think with the time that we have left, 5 minutes, perhaps we can just give some top tips for students applying for the courses. I know obviously earlier we had a look at what would make a good applicant, but do you have any kind of 2 key takeaways as a source of advice for students making their decisions and applying for one of these courses? Edith, should I hand over to you first?
Click the button, apply today.
Yeah, that's the best advice. My advice would also be honest. Don't try to pretend. Just say exactly what you want to achieve, what you want to learn, what you know and what you don't know. We are not here to judge. We are here to help and to teach you. And it's very helpful if in this process we're transparent. We certainly are going to be transparent. We're going to tell you what is possible and what is not, but it's very helpful if you come forward and don't pretend, but are really honest and say, this is where I am and this is where I would like to get to with your help.
Fantastic. Thank you both. I think we don't have any other questions, but obviously, if you have any questions following this event, please do contact Horia or Edith. I'm sure they'll be delighted to answer any questions you may have. So I just wanted to take this opportunity to say thank you so much both for your excellent presentations. And we really hope that this session has informed or given you an insight into what it is really like to study at UCL. And again, we really hope that we will be able to invite you on campus soon. And following this event, we will have a short survey. If you would be ever so kind just to have a quick look over that and let us know what your feedback about the session is. And yeah, thank you so much both. And we hope you'll have a lovely evening.
Goodbye. Good luck.
Take care, everyone.
Thank you very much. Bye.